I'm based out of Boston, United States. So it's 10 p.m. now on the East Coast, and but it doesn't matter what time it is, because in the last few weeks, every conference that I have attended, there's been so much focus on multi-omics, number one, and precision medicine, and in general, translational research, precision medicine, transcriptogenomic, proteogenomic, all these different kinds of disciplines coming together to get a better idea. And so it's a pleasure to talk to you about how better samples can facilitate better quality data. And thankfully enough, we are at that junction where adaptive focused acoustics can actually enable a lot of the labs. So without further ado, let me get started. So I'm just going to do a little bit of a preamble. Probably it's going to be a little bit of repetitious work for most of you who are working in the domain of precision medicine, but in case some of us are not, or if there are people who are not in that space, it's basically the transition and the translation of this whole world of therapeutic care evolved when people realized that one drug fits all method is not necessarily the best of the pathways. And yes, a certain drug will have benefit for some particular population group, whereas it will have no benefit for a certain other section and adverse effect for a different group of people. So this gave rise to the aspect of specific therapy, or mainly the prescription-based diagnostics, that where every person has a doctor today to go to and get some specific drug combination. And how could we then combine the prescription capability and the diagnostic capability to bring the companion diagnostics into equation, which is essentially in the domain of precision medicine? So in order for us to do that, it is not an easy aspect, because every person, there's gender disparity, there's age disparity, there's other genetic disparity, there is metabolomic disparity, there are biomarker disparities. So there are more differences between us as human beings than similarities, so to speak. So it is very, very complex. And when you think about it, this whole portfolio of precision medicine and critical care, it actually results in the creation of a translational portfolio. So you need to think of, or we are all, people in this space are thinking of multiple different domains that are coming together. So how can we actually develop a certain drug that will be precisely accurate for a certain individual, and any individual who falls in that category where this individual belongs to? So it could be a gender-based specification, it could be an age group-based specification, other diseases. So somebody suffering from colorectal cancer who has a heart disease and also has diabetes is aged between the age of like 45 and 65 and is a man, would get benefit if they take this particular therapy. So that's where the world is now heading. So there are, of course, it requires thorough blood phenotyping, identification of the common drug targets, so that the biopharmaceutical companies can actually make the therapeutics meant for those particular drug targets. So that would require people to understand the gene profile, the proteome profile, the transcriptome, microbiome, metabolome profile of this group of people. And then last but not the least, understanding how to conduct the clinical trials. So gone are the days of where clinical trials used to be only monitoring the PK/PD curve. In precision medicine, they are doing platform trials, which is basically a combination of prescription and diagnostic tools coming together. So you monitor the patient while you're treating as well. And it's not necessarily only monitoring the drug that you are giving the patient, because you have to also monitor if the tumor that you're trying to get rid of is shrinking in size over a period of time. So, needless to mention, all this is happening because multi-morbidity types or different kinds of morbid conditions contribute to clusters in precision medicine. So if you can see in the graph shown here or in the pictures shown here, the one on the top is basically where you have a certain type of cluster, and every cluster has the same set of comorbidities. The second one is multi-morbidity. So, and it's discrete, that they all have morbidities, but they are different from each other. So you can see there are 3 different groups. And in the last one, the lowest one, you are seeing that multi-morbidity is in the causal pathway with the clusters. So basically, it is giving rise to certain other issues, such as causing phenotypic variation and also the epigenetic changes. Now, what that means is that the precision medicine needs to scale up. And the scaling up thing is not necessarily in terms of the number of drugs that are being made. It is actually the number of targets the precision medicine needs to identify. And the number of cell types, the number of organs, the number of tissues that has to be taken into consideration, analyzed really fast to understand that for us to actually cater a certain individual who belongs to a certain morbidity cluster can actually get help from this. So over here, you can see that, while people are at the biotherapeutic level, at the biopharma level, people are monitoring millions of patients. The GTPM essentially stands for genome, transcriptome, proteome, and metabolome. Now, all of these factors are taken into consideration over a time course across all the affected organs. So you can start with a single cell sequencing, then you sample in multiple different organs, then you go and monitor the same gene that you expect to be expressed in a patient who's having all those different kinds of diseases together across other millions of patients. That is this particular gene or proteome or metabolome present in that individual, because that becomes eventually a marker. And then designing a therapeutic pathway that can cure or suppress or downregulate that proteome, that metabolome, that transcriptome, that genome. So needless to mention, in order for us to do this, there is some significant requirement for us to have thorough knowledge of the genomics, transcriptomics, proteomics, and metabolomics, because we need to know how these areas function before we actually get into the domain of figuring out how does it function, how does the genomic profile look for someone who's suffering from some sort of a cancer. If we know how it works, and now in today's world, after the Human Genome Project, we know how the human genome looks like, so we can figure out what the oncogenes are that has resulted in the formation of so many library preps, so many assays, resulting in the next-gen sequencing laboratories. Same thing, is happening with the proteomics-based treatments also. There are so many proteomic markers that are being routinely used in some specific diseases. So I really like this statement from David Wishart, who's a professor of biological science and computing science at the University of Alberta, Canada. And he starts by saying, we are nothing but a bunch of chemical reactions. And indeed we are. I raise my hand, there is a phosphorylation happening somewhere in the body. We have so much of chemical reactions happening in our body every morning when we get up and have our breakfast, when we have lunch, when we have dinner, and even when we are sleeping. Our basal metabolic rate is taking care of so many chemical reactions. So we are actually a product of God knows how many chemical reactions that are happening every second, every millisecond. So in order for us to do this, the biggest issue at the laboratory level, now let's come down from the high perspective down to what do we need to do to implement a system where we can actually have very good idea of the genomic profile, the transcriptomic profile, proteomic profile, metabolomic profile of a patient, of a person, of a population, so that we can design a better drug, a better vaccine, so on and so forth. So first and foremost, knowing the disease type, and the most important thing at the analytical level is basically ensuring sample quality. That's where we start. We have a tissue, we have some cells, we have organs, and that's where we are starting with. And whatever extraction processes that we are using today, we are continuing to use it with the hope that it is actually going to give us a much better data. Now, it won't if we are not making any significant changes. And so our challenges in the laboratory continue to grow. We always look for better data. So more robust, higher reliability, higher reproducibility. We are looking for ability to address more complexity in samples. We are always looking for reducing cost per sample. We are always looking for increasing the level of complexity of analytes. So if we, in a proteomic world, if we could identify 2,000 proteins today, can we get to 4,000 proteins tomorrow? Can we do a holistic portfolio, like genomic, transcriptomic, proteomic, metabolomic, whole profile from one tissue that's divided into 4 different parts? Can we get a full profile from one tissue sample, from one scroll? And all of that done in a really fast pace within a short period of time. Now, there is no magic key that can open the lock to this world of mystery. There are some magics there, however, that can help you address some of the challenges. And I would not say that I'm here today to say that, oh, adaptive focused acoustics is going to help you address all of them. But we surely can help address a whole bunch of them. So first and foremost, I would like to introduce what this adaptive focused acoustics is. Essentially, it's an acoustic energy. It's a technology that delivers controlled acoustic energy that addresses the bottleneck between the initial sample collection and downstream analysis. So what it does, basically, you have your tissue samples, you have your cells, you have your organoids, and you put it in a tube or a vial or in a plate, and the acoustic energy goes and hits the sample, which is residing in the well of a plate or in a tube or in a vial, in a buffer. And it creates millions of cavitation bubbles because of this energy and because of a defect that is being introduced into the consumable. That's the tube or the vial or the plate that you're using. So as a result of this cavitation bubbles, what you can see at the top part of the screen, that you will see that, because of this energy's ability to contract and expand. So this alternate contraction and rarefaction results in eventually implosion of the bubble, thereby creating a shearing energy that disrupts the tissues, lyses the cells, disrupts the organs, and releases the analyte. Now, that's not all there is to the story, because you might be saying that, oh, that is exactly, I expect a sonication to do something similar. Yes, but your typical sonicators are either going to provide too much of pressure, which you're seeing in the bat sonicator. Probe sonicator is not bad, but when it comes to heat and contact, that's where contamination and excess heat jeopardizing your sample, sample integrity. Now, that means that adaptive focused acoustics is actually delivered in a non-invasive way, which means it is not touching your sample. It's coming from below through a water bath, so it's coming under very isothermal or temperature controlled situation, and it's coming from below, so there's not a single probe or anything that touches your sample. So there's no contamination, hence non-invasive. The temperature is retained at the same level because it's coming through a water bath. As a result of which, you deliver this high impactful energy that helps you extract most of your analytes, whether it's DNA, RNA, proteins, metabolites, doesn't matter, get you very high quality and high yield of these analytes from samples, and without jeopardizing their integrity. So it's not necessarily that I'm going to say that this is the best, you have to accept my comments as that this is the best thing on the face of this earth after sliced bread. I'm not going to say that, because already a lot of people say that this is a wonder in the world of sample prep, which is why it is considered to be the gold standard of sample prep for almost every next-gen sequencing laboratories. So my purpose of showing this slide is that from tissues or cell lines, from chromosomes, all the way to messenger RNA, you actually go through the process. That's what people are doing, where they are extracting DNA and RNA from tissues and cells, shearing it down, and then doing amplification, whether in using an RT-PCR or they are using the messenger RNA or RNA for some other studies. And this is being done extensively in today's world. And I'm going to show you some examples of that. Now, what we are seeing as a development in the last 5 to 6 years is this humongous evolution in the domain of proteomics. And now we are seeing the evidences of metabolomics also coming into play. Because one of the main reasons, as I mentioned before, that every single genomic lab that is moving into the precision medicine is also needing or asking for data from the proteomic metabolomic perspective. And every single proteomic research group or metabolomic research group is also looking for genomic alignment and agreement with the data that they are seeing. So there's top-down and bottom-up requirements from all parts of the world are becoming so important that everyone now needs to know all that there is. They just don't want to get to know that, oh, this gene, oncogene, is present, and that means what? Does this person also have these particular proteins upregulated? Is he making any of these metabolites? Because if he does, then for a patient who has these oncogenes expressed and has a higher concentration of this RAS and Rho proteins and certain metabolites, should undergo this particular anti-cancer treatment regime for a faster recovery or lifetime extension. So what I'm going to do today is basically take a look at how AFA works with different matrices, how it works with different pathways, and how it works with different application types, like NGS, like proteins or protein analysis, so to speak. So first and foremost, I know I need to speed up a little bit, but because I definitely want to finish in the next 20-25 minutes so that we have time for Q&A. So this is an example of different cell lines, and you can see the total amount of cells from different sources. So you have the lipolytica, which is highly hydrophobic. You have whole blood cells. You have epidermides, which is very, very hard cells. And then you have the Cerevisiae, E. coli, so on and so forth. So all of these cells were lysed using AFA at certain specific conditions, and all of these lyses was achieved in less than 60 seconds. And the goal was to actually get to see how much of DNA and how reproducibility could we get to that 40 kilobase DNA within 60 seconds. And needless to mention, you see from the reproducibility graphs that you get consistently across all cell lines very high amount of yield just within that specific time of AFA or with focused acoustic energy. Now, it's not just the DNA. You can go and also look at cell lysis looking at RNA from yeast. And you can see over here that you are going to get intact ribosomal RNA, as well as you get higher yield and shorter duration for getting to that higher yield of RNA. So basically, you can monitor the RNA quality in terms of the DV200 scores or the RIN numbers. I always prefer the DV200 scores because RIN numbers are not necessarily always very specific. So there are a bunch of application notes that we have published thus far. I have shown a couple over here where we are looking at, low volume genomic DNA extraction from whole blood or lysosome extraction of biomolecules or DNA and RNA, especially from eukaryotic and prokaryotic cells. And moving on to a little bit of my favorite domain, which is proteins and metabolites. Here are some examples of where proteins from E. coli were, overlapped with each other in terms of showing how reproducibly they were achieved from different cell lines, different E. coli strains that we looked into, and also how reproducibility, we actually extracted both metabolites and specific protein markers to the right of your screen. And there's been a lot of publications also in that space. I have mentioned the eukaryotic, prokaryotic cells and RNA extraction from yeast, high molecular weight DNA extraction, and purification from whole blood. So there's a lot of these things that are already out there. The next one that I'm going to go into is basically introducing you to the world of where we are actually very, very effective and impactful is in the world of FFPE. Now, every person who works in the domain of oncology would know how important FFPE is. And they would also know how difficult it is to work with FFPE because you see your tissue embedded in a block of wax. And you take a scroll and you spend a humongous amount of time in getting rid of that paraffin. Before even you get to the tissue to extract your DNA and RNA, you have probably lost some of your sample and a lot of hours in trying to get rid of the wax that was covering your tissue. So interestingly, AFA is the only technology that can emulsify wax in a matter of seconds. So you do not need any xylene, you do not need any mineral oil or any other kits other than the hydration buffer with acoustic energy to deparaffinize, which we refer to as active deparaffinization. So earlier this year, we actually launched a workflow with very new generation chemistry where we can actually enable somebody to extract not only the tissue from the paraffin from the FFPE block, but also extract DNA and RNA in high purity and quality all at the same time. So something that you would be spending 16, 18 hours to extract is now happening in 5 hours, 5.5 hours, so on and so forth. So here is an example of the workflow where I'm saying that, you can see that, at the top, just, rehydration buffer and deparaffinization gets rid of your paraffin. And then you centrifuge and aspirate the supernatant, and then you incubate it. And this is where you separate the RNA from the DNA. You treat it with you decrosslink or reverse crosslink, and then you get your pure DNA from the pellets. And for the RNA, it's in the supernatant, and you also do the reverse crosslink, and you get purified RNA. We are not just providing the instrument, which you can see on the screen over here. This is an instrument that has the capability of taking in a 96 well plate. We also provide all the consumables, the whole workflow. And if you are into a liquid handler system, we also work with you to optimize workflow on your liquid handler system. So we have the whole workflow, and here are some examples of data where you can see that we are looking at 4 different tissue types: brain, breast cancer tissues, colon cancer tissues, lung cancer tissues. And so we are looking at, so the same kit that I talked about in the workflow is available for an automation system as well as for to be done manually. So the dark blue and the light blue bars are the automated system and the manual system respectively. And we are comparing it with one of the competing kits that are out there in the market. And you see that you get a much better yield from the performance of the auto and the manual kits are one and the same. And they are together always better than that of the competition. In terms of, not just in terms of RNA yield and purity, it's also in terms of RNA amplifiability and molecular weight. So you can see the DV200 scores over here across 4 different tissue types. Same thing you can see about the DNA yield and the DNA amplifiability and molecular weight that you can see. Once again, the kappa score as well as the molecular weight by smear analysis. And then another thing that I always like to highlight is what happens when you get the DNA. You have to shear it down, right? Because you want to know what the gene is. And you have to shear it down to a specific size. And then you will use your RT-PCR to amplify it. And then you look for, you use your library prep to monitor it. So here is an example of purified DNA obtained using our workflow versus something that's by competition or using a competitor kit. Now, at this level, the DNA extracted brain tissue, and we are looking at 4 different sample types for each. They are very similar. But the moment you do the shearing and DNA sheared down to 175 base pair target, you see how our workflow, the Covaris AFA oriented workflow, gives you highly reproducible data versus tremendous disparity and high variability that you see from other kits. So one of the other reasons of thinking about using adaptive focused acoustics for FFPE, all your FFPE requirements and needs is not only do you save time upfront in getting your tissues out of your paraffin block and solving your whole emulsification nightmares, but it's also the quality of DNA and RNA that you get. And that's where the chemistry that we use in our reagents and consumables come into play. So I'll switch gears a bit, moving more to the epigenetics or epigenomics part, which is, as I mentioned, that, in the precision medicine world, you cannot just stick with genomics. You have to look for the transcriptomes, you have to look for proteomes, and you have to look for metabolomes. So I'm going to move into the transcriptomes, and you know that there are several chromosome territories and chromatin territories. The transcriptional machinery, the transcriptional factors, chromatin modifiers, and then you monitor the histones and do a lot of those things. So at the end of the day, if you are in the world of epigenetics, you bring in your sample and you need a workflow that is actually very, very versatile. So you need to actually have a method that is efficient and preserve the sample from degradation. So that needs to be in the sample prep domain. And then you preview the information by monitoring or having to optimize your capability which is dependent on what you are trying to study. That is, are you looking at large RNA protein complexes versus certain histone modifications, histone markers? And then the next part is looking into the extracts. So you need to have better quality of your extracts and defining the decision window, or where you are looking at the molecules, where you are detecting the RNA protein complexes or even the histone modifications. That's where you need precision. That's where you need reproducibility and reliability, because the life of the patient is depending on how you are doing it. So the process needs to be user-friendly, and the process needs to be precise, and it needs to be compatible with the different other things, like different NGS platforms, different mass spectrometry platforms, and automation systems, and so on and so forth. So as part of our chip, Chromatin Immunoprecipitation, we have this solution that covers some of these critical first steps of the workflow that actually works very well with sample prep, or does an extremely good job when it comes to sample prep part. So Covaris systems have been very instrumental in many other challenging chip-related methodologies. So we have enabled the Sharp-seq, the High-seq, the ChIP-seq for sure, the Methyl-seq, the Kia-PET, and so on and so forth. So what we do, once again, to reiterate, I'm not going to say, like I said at the very beginning of the presentation, that the adaptive focused acoustic energy is not going to give you answers for everything, but it can give you very good quality samples, starting from your complex matrices, so tissues, organs, cells, any other sort of complex matrices, and your plasma, whole blood cells, so on and so forth. So that whole sample preparation process, where you need to cross-link the proteins and then lyse the cells, and then you have to do the shearing of the chromatin, that is where we actually come and play a very critical role. And what is typically done as a method of choice for nucleic acid modification study is that you have the bisulfite conversion, which is also a part of the bisulfite sequencing. And you do that by converting a cytidine to uracil because of the bisulfite conversion, and you have the methylated cytidine. So when you do that, then you just amplify based and monitor it with your library prep and NGS sequencing. Now, doing so with our capabilities, actually, one can see, as you can see in the box over here, is that you get a much tighter distribution of the fragment sizes that helps you to an easier data interpretation. And if I go a little beyond to protein DNA interactions, that's where, DNA is the information carrier, as we are all aware, and proteins progressing our lives forward. So the interaction between the DNA and the protein have some critical importance in the developmental phase. And an important mechanism of protein DNA interaction is via direct binding. And what we do in this particular case is our truChIP capability or workflow offers this unique approach to chromatin shearing. So right at the very first, in the blue box, you see in the sample preparation, you start with your sample culture, and you can actually look at your cell density, fixation of the cells, lysis, nuclei prep, chromatin shearing. All of those cases factors are taken into consideration. Then in your immunoprecipitation phase, AFA also enables, or our workflow enables, antibody choice and IP buffer composition. And then in the purification, you have this ability to actually do the reverse cross-linking. You can also do the RNase proteinase K, and you can choose your column or magnetic bead use, so on and so forth, and then you do the analysis. So where our truChIP, which is one of our kit and workflow, and AFA comes into play is that we offer all this in a very robust, comprehensive, easy to implement manner that can work in the front end with your samples, resulting in chromatin preparation with truChIP, chromatin shearing with adaptive focused acoustics. You can capture the protein DNA complexes with our tag or with the IP kit, and then you can get your purified DNA for your library prep, resulting and eventually getting into high throughput sequencing. And so, what exactly are you going to get with Covaris? So essentially, it's the reproducibility of the data. So you get robust system, reproducible and easy to use. You get a very easy to follow intuitive protocol, and you're going to get your correct sizing post DNA shearing. You can preserve all the epitopes, and your shearing will not have any of the GC biases. So all the AT-rich sequences will be preserved, and also it's going to be highly, highly scalable depending on your requirements. So essentially what I am highlighting over here is we have had better sample prep for confident results across a host of matrices for different analyte types, and looking into different applications, whether it's NGS, whether it's LCMS, ELISA. And I'm going to switch over to some of the LCMS data now. So just to refresh our memory, in the domain of oncology, we have the ability to actually look at from a genomic perspective. So once again, genomic, transcriptomic, proteomic, metabolomic, GTPM, the 4 building blocks of precision medicine. We have the ability to work with any of the genomic requirements, so we can address from 150 base pair DNA all the way to 5 kilobases in terms of shearing, with high reproducibility that you see in the middle right of the screen, and then also high yield of DNA that you can see. So once again, that is why we are considered to be the gold standard in DNA shearing for NGS. Similarly, when it comes to in the domain of oncology, RNA shearing with AFA, whether you are looking at exclusively messenger RNA or total RNA, you can look at the difference of the two data when you are comparing unfragmented control and with 80 seconds of AFA processing, how controlled your RNA shearing would be. This is messenger RNA, and this is total RNA. So you can see the difference. And then, you are not going to be landing with 2 different types, 2 different sizes. You are going to have one high concentration of specific RNA size. So AFA is known to lower library prep cost because it is reproducible, and it's reproducible over a wide range of size selection. So necessarily, or you do not necessarily need to keep yourself only within a specific size for RNA or DNA. You can have a wide range, and you can work with broad range of DNA concentrations, and it is still very, very cost efficient, as was reported by this company, Genosity, that's one of our users, where they are seeing like 10 cents per sample of expense using AFA versus their previous expense of $6.40 per sample. Now switching gears slightly into the protein world, and because I've covered the genomic part, including DNA and RNA, and the transcriptomic part also highlighted some of the epigenetic, and now I'm going to go into proteomic area. So this is where things start getting complex, because until now we had 4 subunits, and now we are going to work with 20 amino acids, and you can have more than 20,000 different proteins in the body, and their functional diversity, and different groups, different labs are focusing on different things. So some clinical diagnostic organizations are looking at biomarkers, whereas some biotherapeutic companies are going to look at monitoring their biotherapeutic that's been injected into a patient only, and they don't care about anything else. any other proteins, whereas the proteomic researcher is going to look at the global proteome. So enabling sample preparation for protein analysis, it is actually what we do the best is somewhat captured in the slide here, but it is much better explained in the following slide, where you can see that it's a one plate capability that takes your tissue, disrupts it, exposes the proteins. You can then use the pull down, sp3, strap, anything you wish, in the same plate to pull down the proteins. And then wash it off. Then your bound proteins can be digested also in the same plate, resulting in finally getting your signature peptides that you're going to monitor eventually with your LCMS. So from adding the tissue all the way to getting your samples LCMS ready, everything can stay in the same plate. Whether it's an 8-strip or 96-well plate or 384, it's completely up to your choice, but you can facilitate the whole thing enabled by this adaptive focused acoustics energy using the same plate. So there are different examples to consider. One is phosphorylation of proteins, which are very difficult to capture because they're happening all the time and they're highly labile. Understanding post-translational modifications, which is basically any proteomic and biopharma, biotherapeutic scientist's nightmare or main goal, getting purified proteins, and also doing a sufficiently robust and reliable cell lysis to get all your proteins in. So if I just want to go into some of this protein phosphorylation, excuse me. So what we are seeing over here are 3 different tissue types. So remember, I showed multiple cell types, 4 different tissue types for the FFPE. You can use the same conditions for proteomic analysis all as well, because your focus points are changing here. You're not interested in the DNA and RNA, you are interested in the proteins now. And what the organization, this is done in collaboration with Johnson & Johnson. So what they're trying to do is essentially looking at different ability to disrupt the tissues to get a maximum concentration of proteins come out. Now, remember, this technology allows you to change the power with which you are hitting your sample. So that also means that skin, which is a much harder tissue type, will require more power than ileum or colon. And as such, you can see that for ileum and colon, you can get a much higher concentration of proteins at a much lower power than skin over here, right? And you can see the same example when you are looking into microRNA bound to a certain specific protein. So you can see that, looking at the GI tract, as well as the heart and liver, different power conditions result in different quantities of proteins released in those specific tissue types or cell types, in this case, tissue types. So complexity of matrix resolved. The next question that comes into mind is when you're working with proteins, one thing that you are always worried about is how long you are spending in preparing your samples and running the experiment, and whereas you should be spending more time in optimizing the experimental conditions or doing data analysis even better. So here's an example of doing TMT analysis for the LCMS aficionados. They are all aware of what TMT is. So it's the process of tagging your proteins so that you can monitor them in an ocean of proteins. So typically, that whole thing takes hours to days, whereas this one, if the topmost traditional pathway of looking at cell lysis, reduction, alkylation, cis alkylation, disulfide bond reduction, protein cleanup and protein digestion, peptide cleanup, all these things take about 50 different steps, 40 hours of work. Whereas with the AFA, you are down to 24 steps and it takes approximately 1.5 to 3 hours. So you are 13 times faster than the traditional process. So does that mean anything? That means you are going to be done 13 times faster as well. So you can spend more time in data analysis, data interpretation, and you can actually do more samples at the same time now. So especially if you're looking into automated data analysis or targeted data analysis, which has got, softwares have become so much more advanced in today's world. So the same thing with FFPE workflow that you can find today as well. You can do proteins. Here is an example of FFPE scroll samples, and everything can be done. You can, just like the FFPE workflow for DNA and RNA, we also have an FFPE workflow for proteins in a single plate, and where you can actually do the deparaffinization followed by tissue disruption, and then treating your proteins, digesting your proteins, on-beat digestion in this case, straight away shooting it to an LC-MS system. We have published a paper with Matthias Mann's group where he developed something called AFA-assisted protein aggregation capture, which is called APAC. It's out there as a publication as well in molecular systems biology. And you can see over here, this was also done with a fresh or frozen tissue where they used an adaptive focused acoustic energy to actually automate the whole process of disulfide bond reduction, cis alkylation, protein pull down, sp3 beads addition, protein binding, digestion. Everything was automated using an Agilent Assema Bravo, and then eventually going into a nano LC connected to Q-Exactive high resolution accurate mass. And this was also published. We have also published this as an application note. So going into almost, the last phase of my talk, we can also work with, cell sorters, LCM samples, laser capture microdissection samples. All of those, we have 384 well plates that can take in all these small tissue samples and process them. If you want to pool, you can pool them, or you can actually process them individually. You are going to get sufficient quantity of samples regardless. So we have, over the years, we have developed multiple workflows for protein analysis as well. And that includes whether a single pot extraction using sp3 bead cleanup. We have FFPE scrolls working with sp3. We have FFPE scrolls working with S-trap, and then automated collection and processing of laser capture microdissection samples as well. So we have a humongous line of products that has evolved significantly since the inception of the technology and the company, which started in 1999. And in 2021, we have more than, I think, 12 or 15 different product types, and different consumables and reagents with different levels of compatibility, scalability, application area, different cell types, and so on and so forth. And you can see in the bottom graphs that how our install bases have gone up, how our revenue has gone up over the years, just by enabling better sample quality. And our list of critical customers, KOLs, continue to grow over the years. And it's not necessarily just combined or confined within these names. There's a lot more: the LabCorps, the Quests, the Cedars-Sinai hospitals, and so on and so forth. So when it comes to, once again, to precision medicine, the GTPM, genome, transcriptome, proteome, metabolome, requires high quality samples at every single stage. And it may not be the same laboratory that's doing all of them. It might be a conglomeration of 4 different laboratories. But every single laboratory can benefit from better sample preparation, better sample management to get to high quality, confident data. And that's where adaptive focused acoustics come into play. So having said that, thank you so much for your patience. And patiently listening to me. We have about, 9 minutes left to the hour. So at this point of time, I would hand it over to Seek and see if there are any questions that I can answer.